So I'd just really like to welcome everybody to this second session of the Early Career Researchers Seminar Series that's hosted by the Cambridge Reproduction SRI. And first of all, I really want to thank the speakers that are speaking today and the others that have signed up to do it later in the year. And also to the team of coordinators, Daphne, Tara, Ellen and Grace, who have put this together. So just to explain a little bit about what the SRI is, it's an interdisciplinary initiative that brings together researchers from all across the university who have interest in any aspect of reproduction at all. So everything from science, technology, medicine, arts and humanities. Okay, so I'm just going to introduce now Staša Stanković, who's based at MRC Epidemiology, and she's going to tell us all about the aetiology of reproductive aging.
Thank you very much for this introduction. I'm very excited to have an opportunity to share with you scientific advances on reproductive aging. And what our group is really interested in is trying to understand how we can follow up all the wealth of genetic evidence that we have to better understand the biology underlying reproductive aging and ultimately suggest and inform on potential prediction markers and treatment targets. So let's put this into context. Reproductive longevity is defined as the time between menarche and menopause. The time frame between these two events has been shown to be critical for healthy aging in women as well as fertility. Natural menopause happens at around age between 50 and 52 years, and it is the time when ovaries become depleted of functional follicles. Not only their quantity changes, but their quality or their genetic integrity decreases too. So why is all of this important? Well, more women are choosing to delay childbearing to older ages, resulting in increased demand for reproductive assistance and cryopreservation to prolong reproductive longevity. And as modern fertility treatments cannot fully compensate for the natural decline of fertility with age, it is very important to consider the regulators and physiological mechanisms involved in reproductive aging. The story goes really to the stage of female fetus. A finite number of oogonia are laid down in fetal life, after which no new germ cells are being formed. And that is the peak number of oocytes at any point during reproductive life. Oocytes are then arrested in the first meiotic division and may stay in this resting phase for many decades and only really resume meiosis just before starting to ovulate. However, already at about 16 to 20 weeks of pregnancy, this number starts declining, where oocytes are lost due to the cell death. And this is really an irreversible process, which leads to a population of about 2 million primordial follicles at birth and around 400,000 at menarche. This gradual decline in the ovarian follicle pool due to the cell death and atresia is really continued during the whole reproductive lifespan, and it is associated with reduced fertility in the mid-30s, which is almost 10 years before the symptomatic indicators of menopause start, irregular menstruation around mid-40s, and then ultimately follicle exhaustion and menopause in early 50s, when the numbers have dropped below 1,000. And the normal process of reproductive aging varies considerably among women, so everyone's reproductive curve will look different, yet for all of them is an age-specific decline. And this can be due to the differences in the starting point or how big our ovarian reserve at birth is, but also due to the differences in the levels of this drop here, or the rate of follicle loss. In less common cases, which is this left tail of the population curve, these timings are disrupted. And epidemiologically, about 10% of women undergo into early menopause before the age of 46, while premature ovarian insufficiency, which is defined as an extreme form of early menopause, affects about 1% of women under age of 40 and 0.1% under age of 30. And the challenge really lies in the possibility of identifying women with a reduced reproductive lifespan at such an early stage in their lives so that we can take inadequate actions. It remains questionable whether this can be achieved by any endocrine or imaging tests that are currently in clinical practice. And this, for example, includes follicle stimulating hormone, anti-Mullerian hormone, because these markers will really only record changes in ovarian function that have already taken place. So preferably, we would like to base the test on the age at which women will become menopausal, which would then open up the opportunity for any young woman to be tested for reproductive expectations rather than the current situation. And it is likely that we will arrive at that position by combining endocrine imaging and especially genetic information. And really, it is the combination of your genes and the environmental factors that will determine where you are in this population distribution curve. However, until recently, we did not know what those genes are and how genetics and environment interact with each other to shape this population distribution curve. And a number of studies have been performed that partially reveal this genetic complexity. So basically, the left tail or individuals with much earlier forms of reproductive timing were mainly explained by targeted DNA sequencing, which captures these rare variants with a major effect. While the rest of the distribution was studied by large-scale genotyping methods of those common genetic variants with smaller effect. And they are believed to most likely interact with environmental factors more than the other ones. So even with these advances, we are far from understanding the full picture of genetic architecture of menopause timing. So what are the questions that our group and my PhD is trying to answer? We really want to understand the genetic architecture that underlies ovarian aging and the potential to implement genetic tools to identify individuals with abnormal reproductive aging with a final aim to inform their health and reproductive choices. Secondly, we want to understand what are those biological mechanisms that govern ovarian aging and determine the timing of menopause. And lastly, we want to know whether we can use genetics to understand the processes that link menopause with broader health and disease risks in women. And we were very lucky to have great access to data sources that enabled us to perform much more powered genome-wide association study, or GWAS. So what GWAS is, just very briefly, it utilizes very small differences or base changes in the DNA between individuals, or so-called single nucleotide polymorphisms, or SNPs. And we can type the individuals based on these base changes and then compare the frequency of these two different types of base in relation to the trait of interest to identify the ones that pass genome-wide significance and thus defined as having the ability to modify the susceptibility of the trait. Why is the identification of these variants so important to us? It is because by applying different complex methods of annotation of these variants, we can identify genes that are tagged or captured by these variants. And of course, this is just a simplified picture of how the method works. And this finally enables us to use these genes as targets to study complex biological pathways, such as the ones underlying ovarian aging. In our last paper, which will be available at medRxiv during this week and is currently under review at Nature, we studied basically 200,000 women from 3 different studies. We meta-analyzed them. They were all of European ancestry, and we were focused on menopausal age between 40 and 60 years. And from about 13 million genetic variants that we identified, both common and low frequency, we basically singled out 290 independent loci, which were genome-wide significant for age at natural menopause. Altogether, these signals explained about 10% of the variance in the age of natural menopause in the population. And these variants had an effect to shift menopause timing from about 3.5 weeks to 1.5 years. But remember that this is the effect of a single variant, for example. So cumulatively, the effect sizes will add up in an individual to have much more pronounced effect. 10%, is that too little to be useful to make any clinical decisions? Well, we were unclear whether the genetic variants affect these rare forms as well, or the left tail of the population distribution. Right now, it is widely believed that menopause before 40 years is the result of those rare monogenic alleles rather than the effect of many common alleles in combination with environmental factors. So to better understand the contribution of common and low frequency variants, we calculated a genetic risk score for age at natural menopause. And the genetic risk score is basically a sum of all risk alleles carried by a certain individual, which is standardized to a mean of 0 with standard deviation of 1. So the average genetic risk score in the population of individuals which have the median age of menopause would be 0. So we defined 2 outcomes. The first one is early menopause that happens before the age of 45. The second one is primary ovarian insufficiency, or women with menopause below the age of 40. And the third group was all other women in the population distribution. And we really assumed that if it was true that early menopause only happens due to these rare monogenic genetic changes, we would see a genetic risk score of 0 for this left tail in the population distribution. However, the genetic risk score demonstrated that menopausal age at this lower limit of 34 years and upper limit of 61 years had a significant polygenic influence. For example, collectively, women with age at natural menopause of 34 years had an average of 0.5 lower standard deviation than the genetic risk score mean, suggesting that not only rare monogenic variants, but also these common genetic variants act on these clinically relevant extremes of age at natural menopause. We then wanted to evaluate whether our genetic risk score model is capable of predicting which individuals will fall into the highest or lowest category, meaning with what precision we can distinguish individuals with early menopause and primary ovarian insufficiency versus the rest of the population. And the genetic risk score had about 65 and 64% of chance to distinguish between these 2 classes. this is maybe not satisfying to make any clinical decisions. However, as we identify a greater proportion of genetics governing this reproductive aging, our predictive model will improve to be able to make these kind of predictions more precisely. And it is very important to say that our model outperforms, performs significantly better than smoking status, which is the most robustly associated non-genetic risk factor, which had in this case almost no discrimination capacity to distinguish two groups. And this really opens up many questions about whether we think that this prediction will be useful, and more importantly, what kind of societal impact it would have. And as an example, to show you that our genetic risk score was capable to pick up these individuals at the extreme ends of the tail, we basically evaluated that women at the top 1% of the genetic risk score had an equivalent risk of primary ovarian insufficiency as women with FMR1 premutation, which is the leading monogenic cause of primary ovarian insufficiency. So in order to map all these variants to genes, we performed various pathway enrichment tests that clearly point to the importance of DNA damage response processes as key regulators of reproductive aging. And indeed, 2/3 of the menopause SNPs that we identified mapped to DNA damage response genes, some with a key role in DNA repair, and some present across DNA damage response pathways more generally. For example, for 2 identified genes, BRCA2 and BRCA2, we summed all loss of function variants, which are variants with the highest impact on the protein function carried by each individual in each of those genes, and we summed them into a single score or burden score. And we show that women who carry a loss of function in BRCA2, for example, had 1.69 years earlier age at menopause, while if they carry loss of function in CHEK2, they have 2.36 years later age at natural menopause. And what this all tells us is that the rate of decline of the ovarian reserve is largely determined by the genetic integrity of oocytes, and that the variation in cell death after DNA damage response is really, really critical. So we actually demonstrate that experimental manipulation of these DNA damage response genes, which are highlighted by the human genetics with previous analysis, can help improve ovarian reserve either by altering this initial size of the ovarian reserve or its decline. And we really focused on 2 examples involved in DDR. So the first one is the SuperCHEK1 mouse model, which has an extra copy of murine CHEK1. And we show that this overexpression increases the ovarian reserve at birth, as you can see if you compare these 2 peak oocytes number, compared to littermate wild type controls. And not only that, but it also led to a prolonged genomic integrity, enhanced follicular activity, as well as delayed reproductive senescence. And this is really all likely as a consequence of DNA repair processes being active during mitosis and meiosis that decrease the apoptosis rate. As we mentioned before, only 1% of the oocytes are ovulated during the reproductive life, with the majority of them going through these cell death processes along the folliculogenesis. And many factors will define the degree of this fall in the oocyte count, including DNA repair. So in our second model, which is a CHEK2, a transgenic CHEK2 mice where CHEK2 is inactive, we show that the inhibition of this apoptotic machinery can slow the rate of oocyte depletion and results in improved ovarian reserve at around the time of reproductive senescence. And importantly, this happened without initial increase in the ovarian reserve, as you can see in this graph between wild type and transgenic CHEK2 model. And we saw that DDR plays an important role, but we wanted to assess the expression profiles from fetal to adult ovaries to demonstrate that DDR mechanisms could or could not act across the life course of, in order to define the ovarian reserve through different mechanisms. And we did indeed show that this is true, while some of the DDR genes were specific to this fetal stage, while the other ones, such as endocrine and metabolic regulation, were specific to the adult stage of reproductive aging. And these findings are really critical, as improved knowledge of these mechanisms may also allow for their manipulation in humans, more specifically halting or temporizing the process of the wastage of the oocytes. And lastly, we wanted to explore what would be the health consequences of this manipulation at the population level. Previous epidemiological studies show that basically a range of correlations exist with the disease, and this is largely supported by our causal inference analysis, where we showed that each 1 year genetically mediated later menopause increases the relative risks of several hormone-sensitive cancers, such as breast cancer. And then that 1 year later menopause has beneficial effects on bone mineral density, type 2 diabetes, and fracture risk. And all these analyses have led to a number of important insights into the genetic architecture of reproductive aging and the biological processes that underpin it. So the future goals for my PhD and our group is that we really want to direct future analyses towards identification of more genetic determinants that shape this reproductive aging journey in every woman. So not only in European, like in this case, but also in non-Europeans. And this includes, beside the GWAS level analysis, it includes UK Biobank exome data, which have much more power to capture low frequency and rare variants, which were not well captured by the analyses that were explained in these studies. This does not only include genetics, but also other omics, such as metabolomics and proteomics data that are available in the Finland study. Secondly, we want to identify genes and biological processes that can be targeted and manipulated to preserve and enhance fertility. And this really requires functional characterization in animal models. And I think it is a nice opportunity, because we're all in this field, to say that our group is very keen to join forces with other lab-based and computational groups to move the discovery of in field of reproductive aging forward. So please feel free to reach out. I will leave my email at the end of the presentation. And lastly, we want to understand the underlying biology of reproductive aging and the ways it predisposes women to disease risk. We showed the association with type 2 diabetes, cancer, and bone health, but we really do want to go into details into these mechanisms and pathways to deeply understand how these processes work. And lastly, I just want to say huge thanks to everyone from MRC Epidemiology, especially Growth and Development Group, and my supervisors, Dr. John Perry and Professor Ken Ong, but also everyone else who collaborated on this amazing work, going from University of Exeter, Copenhagen, but also other company level participants such as Decode and 23andMe. And as I said, feel free to reach out. This is my email, and we're very happy to set up the collaborations. And thank you very much.